Hey everyone, in this lesson we are going to learn about how gluconeogenesis is regulated. And before I start talking about the regulation, I first want to quickly review the pathway of gluconeogenesis. So first thing we need to know that gluconeogenesis is not nearly the reverse of glycolysis. In glycolysis, one molecule of glucose is broken down into two molecules of pyruvate, and glycolysis has three important enzymes which catalyzes three irreversible reactions. And these enzymes are hexokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase enzyme. On the other hand, in gluconeogenesis, two pyruvate molecules are used to synthesize one glucose molecule. And the three irreversible reactions of glycolysis are bypassed in gluconeogenesis pathway, and these reactions are catalyzed by four different enzymes. One is pyruvate carboxylase and phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase enzyme. So these enzymes actually bypass the irreversible reaction of glycolysis to synthesize glucose, and therefore gluconeogenesis is not merely the reverse of glycolysis pathway. Now let's quickly review the pathway of gluconeogenesis. So in this reaction, the first step is the carboxylation of pyruvate to oxaloacetate, and this reaction takes place in the mitochondria, and the enzyme catalyzes this reaction is pyruvate carboxylase, and this enzyme also requires bicarbonate, ATP, and biotin. Once oxaloacetate is synthesized, it cannot actually cause the mitochondrial membrane, and therefore oxaloacetate must be converted into malate with the help of mitochondrial malate dehydrogenase enzyme, and then malate is now able to cross the mitochondrial membrane to be transported into cytosol, and in the cytosol malate is then converted back to oxaloacetate with the help of cytosolic mitochondrial dehydrogenase enzyme, and oxaloacetate is then converted into phosphoenol pyruvate with the help of phosphoenol pyruvate carboxykinase enzyme, and the reaction uses two molecules of GTP. Now, through a series of reactions, phosphoenol pyruvate is then converted into fructose 1,6 bisphosphate, and in between there are two molecules of ATP which are being used to synthesize 1,6 bisphosphoglycerate. Now, fructose 1,6 bisphosphate is then converted into fructose 6 phosphate with the help of fructose 1,6 bisphosphatase enzyme, and it also releases one inorganic phosphate, and glucose 6 phosphate here then converted into glucose with the help of glucose 6 phosphatase enzyme, and it also releases one inorganic phosphate. And if you remember that mostly in all other tissues gluconeogenesis pathway ends here, however in liver, because the main role of liver is to maintain the blood glucose level, so liver and kidney are the only organ which releases free glucose from glucose 6 phosphate, and glucose 6 phosphate from here can then transported into the lumen of endoplasmic reticulum, and from there the free glucose is being released with the help of glucose 6 phosphatase enzyme. And if you really want to learn about this in detail, please review my previous video on gluconeogenesis pathway. So this is the overview of gluconeogenesis pathway, and now I want to talk about the regulation of gluconeogenesis. Now, the first important thing to remember about regulation of gluconeogenesis is that gluconeogenesis and glycolysis are coordinated so that one pathway is active while the other is inactive. It is important because if both these reactions were to operate simultaneously, there will be no net benefit. In fact, there will be considerable consumption of ATP, and this is prevented by a sophisticated system of reciprocal control which inhibits glycolysis when gluconeogenesis is activated, and when gluconeogenesis is inhibited, glycolysis is activated. Now, the important sides of regulation of gluconeogenesis are three different enzymes. One is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, also known as PepCK, and third is fructose 1,6 bisphosphatase enzyme. And what actually regulates the pathway? First and very important thing is the energy status of the cell, which is if the cellular energy is high or cellular energy is low, and we are going to learn about that shortly. And second is the blood glucose concentration, which is regulated by hormones such as glucagon, insulin, and cortisol. And lastly, it is also regulated by the availability of the substrate. Now, I first want to talk about the hormonal regulation at the interconversion of phosphoenol pyruvate and pyruvate in the liver. Now, this is the glycolysis pathway where one molecule of glucose is converted through series of reactions into two molecules of phosphoenol pyruvate, and phosphoenol pyruvate is then converted into pyruvate with the help of pyruvate kinase enzyme. And the reaction also synthesizes two molecules of ATP. So now consider a situation when blood glucose is scarce or the blood glucose level is low. In that case, the level of glucagon hormone rises, and increase in glucagon level can activate adenylate cyclase enzyme, which can catalyzes the conversion of ATP into cyclic AMP, and increase in cyclic AMP then activate cyclic AMP dependent protein kinase A enzyme. And if you remember that the main function of kinase enzyme is to add the phosphate group on the protein, so protein kinase A inactivates pyruvate kinase by phosphorylating pyruvate kinase enzyme. And because the pyruvate kinase enzyme is now inactivated, phosphoenol pyruvate can no longer be converted into pyruvate, and therefore pyruvate will be then directed or carboxylated into oxaloacetate with the help of pyruvate carboxylase enzyme, and oxaloacetate is then converted into phosphoenol pyruvate with the help of phosphoenol pyruvate carboxykinase enzyme or PepsiK, and phosphoenol pyruvate is then converted into glucose through a series of reactions. So when blood glucose level is low, glucagon level increases, and increase in glucagon level inactivates pyruvate kinase enzyme. That means glucagon inhibits glycolysis and activates gluconeogenesis pathway to synthesize glucose. Now let's consider when the glucose is abundant, especially after the meal, which increases the blood glucose level. So when blood glucose level is high, it increases the insulin level, and increase in insulin can activate protein phosphatase enzyme. And unlike kinases, the main role of phosphatase enzyme is to catalyze the removal of phosphate group from the protein. So when protein phosphatase enzyme is activated by insulin, it then activates pyruvate kinase enzyme by removing the phosphate group from pyruvate kinase, and which then allows phosphoenol pyruvate to be converted into pyruvate, and pyruvate is no longer be able to convert into oxaloacetate and oxaloacetate to phosphoenol pyruvate. So it activates the glycolysis pathway by breaking down glucose into pyruvate. So now you know that low blood glucose increases glucagon level, which activates gluconeogenesis, and high blood glucose level increases insulin, and that activates glycolysis. So this tells you that both glycolysis and gluconeogenesis pathways are regulated reciprocally. Now I also want to talk about how acetyl CoA activates gluconeogenesis pathway. For instance, in adipose tissue, there is increased lipolysis where triglycerides are converted into fatty acids, and these fatty acids are then transported into liver. So liver is flooded with fatty acids, and these fatty acids are then entered into mitochondria as fatty acyl CoA, and through beta oxidation, fatty acyl CoA is then converted into acetyl CoA. Now the problem here is the rate of formation of acetyl CoA exceeds the capacity of the liver to oxidize acetyl CoA into carbon dioxide and water. As a result, acetyl CoA then accumulates, and this increased level of acetyl CoA then activates an enzyme called pyruvate dehydrogenase kinase. Now this pyruvate dehydrogenase kinase enzyme inhibits pyruvate dehydrogenase complex by phosphorylating this enzyme. As a result, pyruvate can no longer be able to convert into acetyl CoA. Now, on the other hand, this increased acetyl CoA also activates another enzyme called pyruvate carboxylase, and because of that, pyruvate is then diverted from glycolysis pathway to gluconeogenesis pathway by carboxylating pyruvate into oxaloacetate. And from here you know that oxaloacetate is then converted into malate with the help of malate dehydrogenase enzyme, and malate is then transported into the cytosol, and in the cytosol malate is converted back into oxaloacetate, and oxaloacetate is then converted into phosphoenol pyruvate with the help of PepsiK enzyme, and then through series of reactions phosphoenol pyruvate is then converted into glucose. Now the glucose, which is synthesized here through the process of gluconeogenesis in the liver, can then be used to replenish glycogen stores. So glucose is then stored as a glycogen in the liver. So the main point here is when the level of acetyl CoA increases, it inhibits glycolysis by inhibiting pyruvate dehydrogenase complex enzyme, and it activates gluconeogenesis by activating pyruvate carboxylase enzyme. Now, one thing to note here that a rise in acetyl CoA indicates that the cellular energy levels are high and that the carbon metabolites can be directed to glucose synthesis or storage in the form of glycogen, as we have just seen here. Next, I want to talk about the effect of energy charge on gluconeogenesis or glycolysis activation. So when I talk about energy charge, I want you to understand that when I say high energy, that means cells have high levels of ATP, acetyl CoA, and citrate, whereas the levels of ADP and AMP are low. And when I talk about low energy, that means cells have high levels of ADP and AMP, whereas the level of ATP is low. Now, based on that, let's say the energy charge of the cell is high. That means there is too much ATP is being generated, then it is being used through glycolysis. So ATP itself can inhibit pyruvate kinase enzyme to inhibit glycolysis process. And alanine, which is an amino acid which can be produced from the breakdown of protein, and this can also inhibit pyruvate kinase enzyme to inhibit glycolysis. Whereas on the other hand, increase in acetyl CoA level, which also indicates that the energy charge of the cell is high, acetyl CoA can activate pyruvate carboxylase enzyme to activate gluconeogenesis. And in the previous slide, we just learned the mechanism of how acetyl CoA can activate gluconeogenesis and inhibit glycolysis. Now let's take a situation where the energy charge of the cell is low. That means there is an increased level of ADP, and this increased ADP level can inhibit pyruvate carboxylase enzyme, and it can also inhibit pepsi-K enzyme. So when cellular energy is low, the pathway of gluconeogenesis is inhibited, and it makes sense because gluconeogenesis consumes more ATP than generates it. So this is the regulation of glycolysis and gluconeogenesis at the interconversion of phosphoenol pyruvate and pyruvate. Now next I want to talk about the reciprocal regulation of glycolysis and gluconeogenesis by fructose 2,6-bisphosphate enzyme in the liver. And most important thing here to remember is that in the liver, rates of glycolysis and gluconeogenesis are adjusted to maintain the blood glucose concentration. So here on the left side we have the pathway of glycolysis, and on the right side we have the pathway of gluconeogenesis. And if you remember from my lesson on glycolysis that fructose 2,6-bisphosphate is an allosteric activator of phosphofructokinase 1 enzyme, and fructose 2,6-bisphosphate is actually an inhibitor of fructose 1,6-bisphosphatase enzyme. So the main point here is fructose 2,6-bisphosphate activates glycolysis, whereas it inhibits gluconeogenesis. And this reciprocal regulation of gluconeogenesis and glycolysis is dependent on the amount of fructose 2,6-bisphosphate available. And these levels of fructose 2,6-bisphosphate is regulated by an enzyme. So for say fructose 6-phosphate here is converted into fructose 2,6-bisphosphate with the help of phosphofructokinase 2 enzyme, which is actually different than PFK1 enzyme. So PFK2 enzyme here increases the level of fructose 2,6-bisphosphate, and fructose 2,6-bisphosphate can be dephosphorylated to fructose 6-phosphate with the help of fructose bisphosphatase 2 enzyme. So this bisphosphatase enzyme actually decreases the level of fructose 2,6-bisphosphate. Now the interesting fact about PFK2 and fructose bisphosphatase 2 enzyme is that these two enzymes are basically one single protein that has two separate enzymatic activities. So this bifunctional enzyme has N-terminal regulatory domain followed by a kinase domain and a phosphatase domain. So PFK2 has a kinase activity and fructose bisphosphatase 2 enzyme has phosphatase activity. Now what activity dominates, whether it is the kinase activity or the phosphatase activity, and that is dependent on the blood glucose concentration. So together, the reciprocal regulation of gluconeogenesis and glycolysis is dependent on the amount of fructose 2,6-bisphosphate, and the amount of fructose 2,6-bisphosphate is dependent on this bifunctional enzyme, and the activity of this bifunctional enzyme is dependent on the blood glucose concentration. And now we are going to look into how fructose 2,6-bisphosphate regulates gluconeogenesis and glycolysis. So for instance, there is low blood glucose level which increases glucagon hormone, and this occurs mostly at night or during fasting. And when the glucagon level is high, it activates this adenyl cyclase enzyme which converts ATP into cyclic AMP, and there is a cyclic AMP dependent activation of protein kinase A enzyme. Now protein kinase A, once it is activated, it actually inactivates PFK2 enzyme by phosphorylating PFK2. That means it inhibits the kinase activity and it activates the phosphatase activity by activating fructose bisphosphatase 2 enzyme. As a result, the level of fructose 2,6 bisphosphate is reduced by converting fructose 2,6 bisphosphate into fructose 6 phosphate, and the reaction also releases inorganic phosphate. So here you can see that when there is an increase in glucagon level in response to low blood glucose, which increases the phosphatase activity, and increase in phosphatase activity reduce the level of fructose 2,6 bisphosphate. Therefore, fructose 2,6 bisphosphate is not available to bind with PFK1 to activate glycolysis. Therefore, glycolysis pathway is inhibited, and decrease in fructose 2,6 bisphosphate is no longer able to inhibit fructose 1,6 bisphosphatase enzyme of gluconeogenesis. Therefore, low level of fructose 2,6 bisphosphate activates gluconeogenesis, and glucose, which is synthesized through gluconeogenesis, can then be released into the blood. Now let's take the opposite scenario where blood glucose level is high, and in that case the level of insulin goes up, and that mostly happen after consuming the meal. And when insulin level is high, it activates this protein phosphatase enzyme, and this enzyme then activate PFK2 by removing the phosphate group from PFK2, meaning that it increases the kinase activity and inhibits the phosphatase activity. As a result, fructose 6 phosphate is converted into fructose 2,6 bisphosphate. So when kinase activity is increased, it increases the level of fructose 2,6 bisphosphate. So here you can see that when insulin level is high, especially after meal, it increases the kinase activity, which then increases the level of fructose 2,6 bisphosphate, which is then able to bind to PFK1 to activate glycolysis. And because increased level of fructose 2,6 bisphosphate also inhibits fructose 1,6 bisphosphatase enzyme and inhibit gluconeogenesis pathway. So this is basically the reciprocal regulation of gluconeogenesis and glycolysis by fructose 2,6 bisphosphate. Now I again want to talk about the effect of energy charge on gluconeogenesis and glycolysis activation. For instance, let's say that the energy charge of the cell is low. That means there is an increased level of AMP, and when AMP level is high, it actually activates phosphofructokinase 1 enzyme to activate glycolysis pathway. Now glycolysis pathway is also activated by fructose 2,6 bisphosphate that we just learned in the previous slide. Now on the other hand, when cellular energy charge is low, AMP can actually inhibit fructose 1,6 bisphosphate to inhibit gluconeogenesis pathway. And in addition, we just saw that fructose 2,6 bisphosphate can also inhibit gluconeogenesis by inhibiting fructose 1,6 bisphosphatase enzyme. So here you can see that high level of AMP and fructose 2,6 bisphosphate, they activate glycolysis on one hand, and on the other hand, they inhibit the pathway of gluconeogenesis. Now let's take a situation where the cellular energy charge is high, such as there is an increased level of ATP and citrate, and this indicates that the cells have too much of energy in the form of ATP, and ATP and citrate, they can actually inhibit phosphofructokinase 1 enzyme to inhibit glycolysis, whereas increased level of citrate, which indicates the energy charge of the cell is high, that can actually activate the gluconeogenesis pathway to synthesize glucose, and this glucose, which is formed here through gluconeogenesis, can then be stored as glycogen. So this is the effect of energy charge on the glycolysis and gluconeogenesis pathway at the interconversion of fructose 1,6 bisphosphate and fructose 6 phosphate. Now I want to quickly review the regulation of gluconeogenesis and glycolysis in one slide. So here you can see that when cellular energy charge is low, the level of AMP goes up, and increase in AMP level can activate phosphofructokinase 1 enzyme to activate glycolysis pathway. On the other hand, increase in AMP level and ADP can activate gluconeogenesis pathway by activating fructose 1,6 bisphosphate and pepsi K enzyme and pyruvate carboxylase enzyme. So when cellular energy charge is low, glycolysis pathway is activated to synthesize more ATP, and gluconeogenesis pathway is inhibited. Now next, when the level of energy in the cell is high, which result in the increase in the level of ATP and citrate, this increased level of ATP and citrate inhibits PFK1 enzyme, and this can also inhibit pyruvate kinase enzyme. On the other hand, the increase in citrate and acetyl CoA, which indicates that the cellular energy charge is high, they actually activate gluconeogenesis pathway to synthesize glucose, and the glucose then can be stored as glycogen. When the energy charge of the cell is high, it inhibits glycolysis and activates gluconeogenesis. And when it comes to hormone, increase in insulin level can activate glycolysis, and increase in glucagon level can activate gluconeogenesis. And insulin level is increased when blood glucose level is high, and glucagon level is increased when blood glucose level is low. Now, quick overview about some of the conditions of glycolysis and gluconeogenesis. So the favoring conditions for both glycolysis and gluconeogenesis. So in glycolysis, the glycolysis pathway is activated when blood glucose level is high, such as when the level of insulin goes up, and glycolysis is also activated when cells have low energy. In that case, glycolysis is activated to synthesize ATP. Now, gluconeogenesis pathway is activated when there is low blood glucose, when glycogen stores are depleted, and when cells have too much energy. Now, positive regulators. So positive regulators for glycolysis is glucose, fructose 2,6 bisphosphate, fructose 1,6 bisphosphate, AMP, and insulin. Whereas positive regulators for gluconeogenesis are citrate, acetyl CoA, glucagon, and cortisol. Now, cortisol I didn't really talk about much, but cortisol hormone is released in response to stress, and when the level of cortisol is high, it actually activates gluconeogenesis pathway. Now, negative regulators for glycolysis include ATP, citrate, glucose 6-phosphate, fructose 6-phosphate, alanine, and glucagon. Whereas for gluconeogenesis, it is AMP, ADP, and fructose 2,6 bisphosphate. Tissue locations for glycolysis are RBC, red blood cells, exercising muscles, and brain. Although glycolysis can take place in many other tissues, but these are the tissues which heavily rely on glycolysis pathway to synthesize energy. And for gluconeogenesis, it is liver, kidney, and small intestine. Subcellular localization, glycolysis can takes place only in the cytosol, whereas gluconeogenesis takes place in cytosol and mitochondria both. Energy used and consumed, glycolysis actually synthesizes 2 molecules of ATP, whereas gluconeogenesis, it does not synthesize. However, it actually consumes 6 molecules of ATP, so it is actually a very expensive process. Now, lastly, I just wanted to make it simple that when cells need energy, such as in the case of high AMP and low ATP, glycolysis pathway is activated and gluconeogenesis is inhibited. When cells have plenty of energy, such as ATP, high ATP and low AMP, gluconeogenesis pathway is activated and glycolysis is inhibited. Now, when it comes to blood glucose, so when blood glucose concentration is low, such as at night or during fasting, gluconeogenesis pathway is activated to synthesize more glucose, whereas glycolysis pathway is inhibited. And when blood glucose concentration is high, such as after meal, glycolysis pathway is activated and gluconeogenesis pathway is inhibited. So this is basically the regulation of gluconeogenesis. And although the title only says regulation of gluconeogenesis, but I think I also talked about glycolysis quite a bit. So I really hope that you learned something new from this video, and I hope that your concepts are clear. And if you really like this video, please like and share the video and subscribe the channel, and I will see you soon next time. Thank you so much.